I am Koben John Nongkynrih, Assistant Professor in the Department of Biotechnology, St. Edmund's College. In this video, I will be discussing in brief about the disease thalassemia. Now, thalassemia is a genetic disorder of the blood. So that means this disease is passed on from the parents to the children through what we call as genes which are present in the chromosome. So that means a child is more likely to be affected by thalassemia if either or both of the parents are affected or they are carrier of thalassemia. So we will discuss in brief later about the genetics of thalassemia. Now let's have a look at the function of the blood and the components of the blood. So the blood has various components. We have the RBCs or the red blood cells, which carry oxygen from the lungs into the various tissues and cells of the body. The second component is the white blood cells or the WBCs. These WBCs, they help to fight against diseases and protect us against infections. Another component of the blood are the platelets. Now platelets help the blood to clot. And the liquid portion of the blood, this is called as the plasma. And inside the plasma, we have the different components which help in the normal functioning of the body. Now in RBC, we have the hemoglobin, which is actually the molecule which is responsible for binding to the oxygen and carry and deliver that oxygen into the different parts of the body. Hemoglobin is made up of two components, the globin, which is the protein part, and the heme group, which is the non-protein part. And in the center of the heme group, we have iron, which is important in the binding of oxygen. Now, the globin part, which is the protein part, these are again of two types, the alpha globin and the beta globin. So these alpha and beta portion of the globin molecules, these come from the genes which are present in the chromosomes. So now we will discuss in brief about the genetics cause of the thalassemia, that is the effects and the changes in the alpha and the beta gene. With the help of this diagram, I will try to explain the genetic basis of thalassemia. As I've mentioned before, hemoglobin has both the alpha and the beta polypeptide chains, that is the protein component. So we have here the alpha and the beta component. Inside these alpha and beta, we have the heme group, and inside the heme group we have the iron. So these iron will bind to the oxygen molecule, as I've told you. So as we can see, we have 4 polypeptide and 4 heme groups. So that's total of 4, and therefore each hemoglobin molecule can bind 4 oxygen molecules. And in each RBC, that is the red blood cells, we have millions of copies of hemoglobin. So a total of about more than 1 billion oxygen molecules can be carried by one RBC. Now, in the alpha polypeptide chain, this part, this is coming from the gene which is present in chromosome 16. Now we have 2 chromosomes, one from the parent, that is the father, and the other one from the mother. Therefore, we have a total of 4 genes for alpha globin. Now, if all the 4 genes are normal, so that person will have normal hemoglobin. If one of the gene, there is change or deletion in one of the gene of alpha, so that person will have a silent thalassemia. He or she will not show any symptoms but is a carrier, and that gene can be passed on to the children. So in this case, we have 2 alpha globin genes which are mutated or deleted, and that can happen either on the same chromosome as in this case, or in this case one on each of the 2 chromosomes. So this case will give rise to mild anemia. And in this third case, as we can see here, there are 3 globin genes which are being deleted. So we have a case which is called as hemoglobin H disease. So this will give rise to moderate to severe anemia. Now, there is also a possibility that all the 4 genes will be deleted. So in this case, there will be no functional alpha globin molecule. So in this case, the body cannot synthesize any of the alpha globin. So this will give rise to what we call hydrops fetalis. Now, this is very severe. It is also called as alpha thalassemia major. Now, in this case, the fetus will die before or shortly after birth because of the non-synthesis of the alpha chains. Now, as we can see here, the 4 genes together are required for the synthesis of the alpha polypeptide chain of hemoglobin. So, if there is a removal of one, it doesn't affect so much the synthesis of alpha globin. 2 will give rise to less production of alpha chain. So that's why it reduces the function of hemoglobin. So that's why it gives rise to the less carrying capacity, oxygen carrying capacity of RBC. And if we have 3, then we have very severe abnormality in the RBC, oxygen carrying capacity. And of course, when there is no alpha globin gene, then there will be that it is not possible to function properly. So the same thing with beta globin. Now in this case, the difference is that there are only 2 genes, that is one on the chromosome coming from the father and one on the chromosome coming from the mother. So if both of these are normal, so that give rise to a normal individual. If one of them is mutation happens in one of them, either on the one from the mother or the one from the father, so this will give rise to a mild anemia. And if both of them are affected, so this will be moderate to severe anemia. So that is the case with the beta thalassemia. So what are the symptoms of thalassemia? So as I've told you, the thalassemia disease affects the oxygen carrying capacity of RBC, and that is the reduce in the number of RBC that is formed. So the symptoms includes some of the similar symptoms as shown by the other types of anemia. So that includes weakness or fatigue, the shortness of breath, then we have dizziness and headaches, and because of the less RBC, there will be a enlarged in abdomen, and of course in general there will be slow growth of the child. And these symptoms, depending on the type of thalassemia that we have seen previously, will show when the child is 1 or 2 years old. Now, if any of these symptoms are present or is observed, it is better to go for diagnosis so that we can confirm whether the child has thalassemia or not. So how can we confirm if a person or a child has thalassemia if we see all these symptoms? So that can be done by performing the complete blood count. This will give us the idea about the amount of hemoglobin, the shape, color and size of RBC. So this will give us whether the person has the anemia which is because of thalassemia or not. And of course we have to cross check with the other parameters to exclude the other types of anemia. We can also go for hemoglobin testing. This will give us the exact type of hemoglobin that is present in the blood, because there are different types of hemoglobin that is present in the blood. And finally, if a child is more prone to thalassemia, because if the parents are the carrier of thalassemia, then we can go for genetic testing even during pregnancy. That is, the fetus can be tested if he or she is suffering from thalassemia. This genetic testing also will confirm what type of thalassemia the child is carrying. That is, what type of mutations happen in the child. Now with these tests all together, we can confirm if he or she is having thalassemia or not, or it may be the other types of anemia, which is of the other causes of anemia. Now we come to the treatment. Now treatment will depend on the severity of thalassemia. As we have seen, if we have deletion in one of the gene, the thalassemia is not shown, but then he or she is just a carrier of thalassemia. But if the person is having mild anemia, then he or she may need blood transfusion. So blood transfusion has to be done based again on the severity of anemia. So through blood transfusion, the amount of RBC will be supplemented so that the body can supply enough oxygen to the different parts of the body. Now frequent blood transfusions will lead to what is called as iron accumulation in the body. Frequent blood transfusion will lead to the accumulation of iron in the body, or what is called as iron overload. So that will give rise to other problems. So this excess iron in the body has to be removed, and that can be done by chelation therapy. So through this technique, the iron will be removed so that it does not cause any other disease in the body. But all of these will not give us a permanent cure for thalassemia. So the only option for a permanent cure for thalassemia is through bone marrow transplant or stem cell replacement. Now this technique can be done so that, because RBC will come from the stem cell, so to remove that defective stem cell, we have to replace that with a good stem cell. But this can be done only if we have a matching donor. So this is the only technique that can cure or completely eliminate thalassemia. So in conclusion, if a child is showing any of these symptoms, so it is always good to go for diagnosis or consult with the doctor so that we can confirm whether the child has thalassemia or not, and if it is confirmed, so that the child can get a good treatment that he or she requires. Thank you.